Welcome everyone to our serial dilution video for microbiology. This is BIOL 2421. The purpose of this video exercise is to show you what a serial dilution is and why we do one. For those of you that are looking on your lab sheets, and I highly recommend keeping your lab guide open at this time, we are looking at page 58 of your lab guide from Blackboard. That is the figure that we'll be looking at. For this, we are going to need our stock water, which we have right here. This is sterile water that I made earlier today. I threw it in the autoclave. The sterile water has 90 milliliters of water in it. It's just sterile water that we got from here, then throw in the autoclave. We'll also need a rack with 6 other test tubes. I have mine right here. Test tube 2, test tube 3, test tube 4, test tube 5, test tube 6, and test tube 7. Now the question, where's test tube 1? Test tube 1 is our source water. Now, the purpose of a serial dilution is to reduce the number of organisms, cells, colonies, bacteria, to a reasonable area where we can count them. A lot of times when we're talking about environmental samples, we don't know exactly how many organisms are going to be on a given surface, or in this case, in a given 10-gram sample of dirt. But we do know that it's probably significantly higher than what we can actually count. So that's really the purpose of a serial dilution, is to reduce the concentration of source colonies, source bacteria, to such a point that we can grow it and be able to count them on a plate. If we did this with raw numbers, everything would just take over the plate, whether it be the actinomycetes that we are trying to isolate, whether it be the bacteria from the NA, or whether it be the fungi from PDA. Again, the goal is to reduce this to a point where we can get colonies, ideally colonies, for counting and for analysis purposes. We did something very similar at the beginning of the semester. Remember isolation streaking? I mean, this is pretty much the exact same reason. For isolation streaking, we want to get isolated colonies, and the reason we want isolated colonies is to see what the colony looks like, its form, how it grows, which you really can't see when it just takes over a plate. Another thing that can happen if you have too many starting colonies, too many starting bacteria, you end up getting the case of what 2 of our bacillus species were, Bacillus cereus and Bacillus subtilis from Biochem Week, in that when it came time to read the plate, you can't really read the plate when the 2 species have taken over the plate. That makes it incredibly difficult to actually identify whether or not it's doing the biochemical response that we were looking for. In that case, for those 2 species, as you saw, we were unable to definitively clock out whether or not, especially with casein and Bacillus subtilis and Bacillus cereus, whether or not they actually did what they were supposed to. By doing a dilution, we're trying to avoid just a complete takeover of a plate. So the basics of a dilution, we have our water, we have our source water, we have our test tubes, we have right here our dirt. This is 10 grams of dirt that was taken from outside and brought in. We also need something to transfer small amounts, 1 mil, from tube to tube to tube to tube. And for that, we have these on our... I will take one of these out. I will not be using this for our dilution. These are sterile, individually wrapped pipetters, and they are pre-measured out to 1 mil. So this will allow me to take 1 mil and transfer it to another. And that you hear in the background is the autoclave. That is our actinomycete auger and our nutrient auger that will be used for the pour plate technique for me later this afternoon, for you guys in the next video. The first thing we are going to do is we are going to combine our sample. Again, this is 10 grams of dirt into 90 milliliters, of water. Give it a nice, snug, tight turn. Now for this, I am shaking it off screen. We want to give it nice shaking so that everything becomes suspended. As you saw, there were bits of tree branches, rocks, and the like. This is because it's not a pure dirt sample. This means that because I went outside, I dug it up and I put it in a bucket, I didn't just get dirt. We get other things, and that is perfectly fine for this technique. By mixing everything up, we're hopefully suspending the bacteria, the actinomycetes, the fungi, into a solution, sort of like that. Now with this, what we're going to do, we're going to remove one of our pipetters. And we're going to transfer 1 ml from here into here. This is test tube 2. Now behind me, I have a vortex. I'm just going to give the test tube a spin. And as you can still see, it's still rather turbid. There's still some color to it, and you can see that there's even some little small specks of dirt and rock in there. For the next stage of the dilution, we're going to take 1 ml from number 2 and move it into test tube at number 3. Again, holding our 2 test tubes. We're going from test tube 2 to test tube 3. I know it's really hard to see. I'm right-handed, which makes it difficult to really see what I'm doing unless I do it like this. And caps go back on. And we give number 3 a spin in the vortex. And we do this again for number 3 to number 4 to number 5 into number 6 into number 7. Now I'm going to show you all of these because at the end I want to show you what the turbidities themselves look like. Again, going from number 3 into number 4. Caps back on. And give it a spin in vortex. Here's number 4 and number 5. Go for number 4 and pipette into number 5. If you notice, it's around here where we actually lose turbidity in the water. We actually can't tell how much of anything there is. It's time number 5, going into number 6. Give it a nice spin. And here's our last one, number 6 into number 7. Number 6 into 7. Give number 7 one last final spin. I'm just going to shift. Now, what I'm about to do is I'm about to take the camera from looking down at the bench top and angling it. So if you have motion sickness, I'm sorry. Now, if we take a look, here's number 1, 2, 3, 4, 5, 6, and 7. And if we take a look, you can see that, again, it's around 4 to 5 is really where our dilution gets to the point where we can't tell that anything is really in there. That doesn't mean that it's completely empty. That simply means that we can't see it. There still may be bacteria, actinomycetes, fungi in the water. In fact, there probably is. Now, if we look at our serial dilution figure, bring this back up. Again, that is page 58. That shows exactly what I just went over, but in a graphical form. It also shows what we're going to be doing for me in a few minutes. But for you guys, whenever you watch the next video, which is plating a technique for PDA, as well as the pour plate technique for the actinomycete auger and the nutrient auger. Most likely, we're just going to be showing an actinomycete demo, since the technique is the same for both. It should be noted that if you had done this in lab, you would have been working with your partner. I am not working with a partner. I am here alone in the lab. So that concludes this video. Thank you very much, and see you for the next one.